now. And also that's my stuff. Okay. All right, so I guess we should get started. Everyone's busy. Thank you very much for taking your time to join us. Our traditionally run ASCO webinar this year. Actually, we would have two. This is part one, which is ASCO 2023 Preview. And we'll talk about moving data, the rise of Asian biotech, and the practice changing trials. We'll get really we're going to go through a lot of contents, but before we start it, we'd like to give a brief introduction for who we are and the panelists. To start with, this webinar is really co-organized by three industry nonprofit. The first one is the Sensory Trust community, which is the youngest, actually, founded by me a few years back. And the second one is BioSpark, which is a Boston-based nonprofit, founded, I believe it's more than 5 years ago. And another one is really the OG, original gangster of I think a Chinese professional organization called SAPA. The full name is Sino-American Pharmaceutical Professional Association. And this is particularly a chapter of Griffith, which is, as you know, is the birthplace of cell and gene therapy. I'm very glad this is three leading, I would say youngest, mid-age, and the most influential, I think, Chinese-focused professional organization co-organized this. And we also get a full media support from some of the leading China meeting outlet. So with that being said, let's move on to the speaker today. I'm very glad to have a stellar speaker today. And I think Lee, she's running a little bit late. I start with John. So John and I actually, like I just mentioned, I started listening to him almost 7 years ago, I would say. Actually, he just told me he started like 8 years ago, right? He started in 2015 with the issue with Mark Shanaban, and then with the Woomer. And if you don't know, their team has been ranked #1 in multiple categories for many years. I think they already lost count. They're just like, they are the analyst team you really want to listen to, because their opinion really changed the market. And I wouldn't say change the market, really inform you the most important data and the most important stone you should turn yourself. That's from John. The second one is Dr. Chandler Park. We know each other a few years back. And I invited Dr. Park, particularly his unique position. Someone speaking, sorry. Oh, a unique position as a high value physician. He's a representative of ASCO, ASH, NCCN. If you really want to know how the drug really impact a regular patient, I would say any of a patient who are treated outside of academic center, you should listen to him. And the third one would be Angus Liu. Angus Liu is a senior staff writer from Fierce Pharma, and also he covered a lot of stuff from Fierce Biotech, especially the company originated from Asia, particularly in China. So he is very experienced, and I believe he has a big, I would say, following in the biopharma industry. So needless to say, we all have our own expertise. Finally, myself, the least one, the last one, also the least one. I'm just, I always call myself a community organizer. I organize the Sensory Trust community. Myself, also a PhD by training, did a few years IO, did have like a little bit sales analyst, a little bit cross-border BD right now, and run my community in Sensory Trust community. Try to bridge the innovation between the East and the West. And in the end, we want to bring the drug to everyone in the world, because we believe disease has no borders, neither should we. With that being said, let's just dive right into the agenda today. I can actually, next page, now turning. This is what we want to talk about. There's 3 sessions, and each they'll lead, I would say, speakers on top of it. We'll start with the market moving data. And I believe I already sent the assignment sheet to everyone, so you already know what you want to talk about. But they're going to be interactive. I guess each each one of us are gonna take a lead with the specific abstract. And then after you finish, if some other people have some question, feel free. And also to the audience, I know we have a lot of people dial in from multiple platforms. And if you really want to do join our Q&A, you have to scan the QR code we offer at some point and join the webinar yourself. Otherwise, you can just you choose whichever platform you prefer. With that being said, I will hand over to John and Angelus. So take it from me, please.
Great. Let's jump right into the beginning. I think by far the most important abstract that came out, although I think it was not the most anticipated data from the conference, the most important abstract to come out was in the TIGIT space. That was Morpheus liver. This is data that everybody knew was going to be in the abstracts, but hadn't been talked up. I don't think people were expecting that this was even going to be the most important TIGIT abstract at ASCO, the most important TIGIT data set at ASCO. But the results are really quite eye-popping. Hazard ratio 0.42, something like that. It's a very dramatic result. Obviously, it's limited patient numbers. It's only a phase 2. And it's worth being said that this is only, the TIGIT-containing arm here is only one arm in Morpheus liver, which has 8 different experimental arms. So this is not the end of the story on TIGIT by any stretch of the imagination. But for a target that the market had treated with a certain amount of skepticism, I think getting this sort of controlled data outside of the lung indication, it should go a long way to convince people that this is a real target. And I think it's getting harder and harder to maintain the opinion that TIGIT as a target is going to be deficient, whatever you think about the quality of the individual studies. Angus, I don't know if you disagree with me on that.
Right. Yeah. I think, John, you mentioned in your note, actually, I saw your note, there's a problem, obviously, in the control arm. I was just wondering if you could go into more details on the control arm. How should we think about that before we head into the phase 3?
Yeah. So look, the problem with control arm is that it underperformed historical control. So you might expect that the most relevant historical benchmark for Atezo and bevacizumab is Embrave 150, and the control arm in the Morpheus liver study of Atezo plus bevacizumab underperformed that, and underperforms that pretty meaningfully, actually. The ORR has a 10 to 15 point delta. PFS, median PFS, has a couple of months, almost 3 month delta. So that seems like a pretty big discrepancy with the historical controls. That is an issue itself, but it's even more an issue because it reminds people very much of Roche's other phase 2 trial with TIGIT, Cityscape, where again in lung the control arm underperformed historical control by a pretty substantial margin. So is Roche just like not running their trials well? Are they not picking the right patient populations here? I do think it's worth pointing out a couple of things. One, it's an internally controlled trial. So both Cityscape and Morpheus liver have an internal control arm, and you can't discount that entirely, right? That's just statistically not fair. What's the point of having a control arm if you're just going to throw it out as soon as you see the data? So you do have to pay some attention to that. Second, the treatment landscape has changed since those initial studies were run, and this is very obvious in lung because we get so many studies coming out with control arms to compare to historical controls. But even just looking at Merck's data in lung as an example, their initial first-line studies of Keytruda had median PFS in 10.3 month range, 10+ month range, and then as data has marched along over the fullness of time, the course of time, that median PFS number has gotten all the way down into the 6s if you look in real-world use of Keytruda studies these days. So over time, we have seen quote-unquote underperformance of control arms that we ought to know what they're going to look like. I think that has a lot to do with what standard of care looks like, how these patients are being treated in the broader context, and what the variety of patients getting enrolled in these studies are. The really high performing patients don't end up on clinical trials like this. They end up getting standard of care treatment in the first line. They do very well. Meanwhile, the early trials, like Embrave 150, had their first choice of patients, and they could select for the absolute best patients to put in the trial. And their inclusion criteria were pretty tight. So maybe it's not a surprise that we see some degradation.
Right. I think, John, I agree with you. I think we're definitely seeing a trend of a decreased performance from compared with some of the IO trials compared with historical control. But I also want to mention the recent LEAP-002 trial, where you have Lenvima, the control arm, outperforming historical control. I mean, if we consider that risk, having a control arm that outperforms, and this phase 2 data being an outlier and outperformer for TIGIT, you could have some very interesting dynamics heading into phase 3 here. So how should we consider that risk?
No, it's certainly a risk, but that's always going to be a risk in clinical development, right? I think we all know that the data never gets better than the phase 2, right? Phase 2 is always the best it looks. Because if phase 2 underperformed what the true value of the drug was, then the phase 3 would never get run. So you only ever see outperforming or seemingly outperforming phase 2.
Yeah.
So I do expect that there's a risk going into phase 3, but I do take heart in the fact that now we're starting to see a pretty consistent delta for TIGIT over internal controls across trials. And maybe the size of that delta is up for debate. But I don't think that it's fair to say anymore that there's no there that TIGIT is doing.
Right. Yeah.
It's worth saying that even if you use the Embrave 150 control arm, TIGIT still looks very active in this.
Yeah, there's still a healthy lead of TIGIT over historical control in the Embrave trial. Yeah, that's correct. That's one difference with the Cityscape skyscraper situation, where you see kind of the TIGIT arm in the phase 2 trial even underperforming historical Tecentriq monotherapy in that trial, where here you have kind of a healthy lead of TIGIT over a historical control.
It'll have to go to phase 3 at the end of the day, right? But I do think it's an interesting, certainly the most unexpected signal in the abstracts that I can think of.
Yeah, definitely one of the positive signs that we're looking for. Leon, I think we're good to move on to the next project.
Let's talk about TROP2. This is a big fight right now, right? I feel like Gilead.
Actually, so by the way, I just want to remind everyone. Sorry, John. So I am just listening and try to assign, the sentiment for each abstract. So my feeling is like you and Andrews agree this is kind of pretty positive. So this is what I'm gonna do. You say I do this. I learned from you, by the way, John. We do this. Yeah, here we go. We got it. So that's you just keep talking. I'll listen, and if I did something wrong, not what you thought, let me know. They're gonna be green, teal, gray, pink, and red. Let's let's say what the color gonna be by the end of the day. So just as a reminder, this is gonna be our prediction, right? Whether say we are good at what we do, or we are pretty horrible at what we do. After I'll ask you, say whether the results really match what we are talking about tonight. Anyway, continue. No, don't worry about me. You guys run the show perfectly fine.
Yeah. Well, the TROP2, it's a big fight, Angus, don't you think? I mean, obviously Gilead seemed like they had this entire space wrapped up. But AstraZeneca coming hot on their heels, and Merck and Kelvin also showing really strong data in these abstracts. At this point, it feels like this space is still very much up in the air.
Yeah, I think, John, to me, just looking at the data, the dynamics among the three players, AstraZeneca, Merck, Kelvin, and Gilead, to me, I think for Merck, the safety advantage should be the key focus here, just because of the lack of interstitial lung disease, especially in non-small cell lung cancer patients, because these patients already have compromised lung. And even in breast cancer, I've talked to several physicians, doctors at top academic centers. They always cite ILD as a major concern, especially in earlier lines of treatment, and that's in breast cancer, not in lung cancer. So I was just curious, where do you see this is heading, especially for the Merck Cullen TROP2 antibody drug conjugate?
No, the ILD signals are really, really important to prescribers, and I think maybe they're underappreciated by the street or by the investor group at this point. This is certainly historically a talks of very strong focus in high grades. But once management of ILD has gotten better and people have gotten better at looking for it, then you start to see the high grade proportion decrease. And once you start to get low percentage grade 3 and above, people stop paying attention to it. But obviously, I think you're absolutely right. Prescribers still pay very strong attention to it, especially for patients who already have compromised lung function.
And John, where do you see the efficacy comes in at this point among the three TROP2 players, AstraZeneca, Merck?
Yeah, the Kelun data looks really strong. I think Gilead data put a pretty strong bar, but AstraZeneca looks to match or exceed it, and Kelun obviously still early data set, but looking very, 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 very strong. I think the trouble at this point is going to be there are some slight differences in baseline characteristics. There are some slight differences in population. So we are going to have to see what those later stage trials and larger cohorts, especially for the Merck Kelun programs, look like. But AstraZeneca clearly is proceeding very aggressively with their trial timeline.
Right.
They've got multiple phase 3s running in all of the major indications. Data there starting to come out second half this year. So it's certainly not going to be Gilead alone in this market for very long.
Right. I think, John, you mentioned a lot about this. I think Merck and Kelun, their TROP2 is a little bit behind in development. And we're seeing AstraZeneca updating phase 1b data from the TROPION-Lung2 trial of Dato plus Pembro with or without chemo in non-small cell lung cancer. So I was just wondering if you have modeled kind of the clinical development timeline for these three assets in non-small cell lung cancer, like how far away is Merck behind AstraZeneca at this point?
I think it's, if you just look at average trial lengths, you can talk yourself into the notion that Merck is maybe even 2 years behind AstraZeneca. But I don't think that's necessarily fair. If there's anybody who knows how to accelerate a clinical program dramatically, it would be Merck. So keep an eye out for those late stage trials getting upsized and getting ramped very aggressively.
Yeah. To me, there's also an interesting dynamics among the three TROP2 players in that, you don't have the Enhertu versus Trodelvy, HER2 low, HER2 negative breast cancer situation where, doctors can discuss sequencing therapy. But here you have all three drugs targeting anti-TROP2, and they have even the same class of payload linked to them. So there is kind of, where do you think we should think about the competitive landscape, just having entirely in-class competitors in the anti-TROP2?
Well, I don't think it's necessarily winner takes all, because I think the data is going to be a little bit tighter than that. Maybe you really do need a pretty meaningful delta in order to take the entirety of the market usually. But given that Gilead is out in front in terms of the commercialization and AstraZeneca coming with what looks like very strong, maybe even better data, but a little bit behind on the commercialization, I do think you'll end up with a split market. But if the phase 3s show differentiation for AstraZeneca, you should expect them to start taking meaningful share immediately and growing share over time. Obviously, early data from the other program is still needs to be borne out in later stage trials. But 44% ORR is very dramatic. If that gets maintained, I would expect to see this as a split market, if not dominated by AZ or
Right. Okay, I think we're good with the TROP2 data. We're just gonna move to the CDK4/6 space. I'm gonna start with this NATALY trial, and John, if you could talk later about that Pfizer asset, the next gen CDK4, that would be nice. So for me, heading into this year's ASCO, this study called NATALY is one of the key studies I'm focused on. This is remind everybody Novartis Kisqali, ribociclib as an adjuvant therapy for HR positive, HER2 negative early breast cancer that's at high or intermediate risk of recurrence. And remember, this is a data set that lifted Novartis stock price by 8% in a single day. After Novartis said it had hit primary endpoint, you wouldn't see that kind of stock movement for a big pharma company very often, they say. So for background, before this data readout, Eli Lilly has already had the Verzenio and abemaciclib approved as adjuvant therapy, but only in high-risk patients based on data from the MONARCH-E trial. There are a couple of differences between the two studies. NATALEE includes intermediate risk patients who had no lymph node involvement, whereas MONARCH-E patients must have nodal involvement. And the second difference is NATALEE uses Kisqali for 3 years, whereas MONARCH-E, the Verzenio is given for 2 years. Now, before this readout, I interviewed Dr. Angie DeMichel, who co-leads breast cancer research at Penn Medicine. She said that that MONARCH-E trial is still, that label is still very limited, meaning there's still need to cover a broader patient population to include the lower risk patients. And by Novartis estimate, the addressable population for NATALEE is about 2 to 3 times that of Verzenio. So, but there is to reach a broader population, there is a problem here, especially for the lower risk disease, because patients would want to see a bigger benefit to justify for additional treatments, because they are already less motivated. They have lower risk, they're less motivated. And that's going to be difficult for Kisqali, because if you look at MONARCH-E, the 3-year invasive disease-free survival is 89% versus control arm 84%. So a 5 percentage point difference. And these are already high-risk patients, meaning they have a bigger room for a new therapy to show some improvement, show some benefits. And when I interviewed Dr. DeMichel, she said that some stage 2 patients, the risk of invasive disease is just 8% over 5 years. So these patients have a totally different mentality compared to some of the high-risk patients. And just imagine that even if you reduce a number by half from 8% to 4%, there is a question about, is it really worth it to put the other 92 patients or 3 years of additional patients just to get those additional 4 patients
Angus,
potential.
What do you think this is going to end up looking like? When
I think just based
in the real world, what proportion of the addressable population do you think will actually opt for therapy?
Right. Yeah, I think essentially this will still initially be the high risk population, just because this is also an interim analysis from NATALEE, and we might not see a full picture of the benefit. And high risk patients, maybe I'm going to be very careful because I've already seen the data, but but still, not to give you any indication, but just even before I see the data, there are also many discussions about whether the high-risk patients will be driving the full analysis. Again, this is not about what the data actually looks like, but there were discussions beforehand. I interviewed Eli Lilly before this readout. Their oncology executive team was that they expect this to read out positively, but it will be driven very much by the high-risk population.
Well, I was about to ask you, maybe it's not fair to ask you if you've already seen the data. I was about to ask you, how do you expect in the high-risk subpopulation to compare versus standard care? Is the delta great enough for this to take care?
Yeah, I did see the data, but I haven't seen the subpopulation. Just to be clear, I haven't seen any high-risk versus intermediate risk, but that will absolutely be a focus of many of the investors and me as well. Whether we will see a difference in the.
If it's not at the presentation, I assume they'll be asked about it.
Yeah, and let's not forget, FDA will also have a higher bar for patients with lower risk. Let's not forget how Verzenio got that initial restriction based on the Ki-67 scores. The FDA obviously wants to see a clearer benefit for some of the lower risk population. So that's also a problem. And we should also consider, obviously, for any efficacy, it needs to be weighed against side effects. Obviously, CDK4/6, this class has various side effect problems.
Well, maybe that's a good segue to start talking about
the next generation.
CDK4. The efficacy in this abstract looked really great, I thought. almost 30% ORR, which, to be fair, was basically as they had guided to the street beforehand. So the efficacy was good, but not unexpected. Safety is always the issue for these drugs, though. We know that all of these CDKs do come with pretty meaningful liabilities there, and sure enough, the abstract does look like the therapeutic index is very tight. I think that's what we said in our note. It's they do not have a ton of room to dose around, right? The efficacy is at 300 mgs, and then 400, they're already running into DLTs, and that's sort of a theme with these. So you don't have much wiggle room. So it makes me wonder: do you have to go? Do you have room to go to combos? Do you have room to really add agents to make these later lines of therapy start to look really good? 30% ORR in a post CDK4/6 population is meaningful, but obviously still leaves plenty of unmet need. And if the dose window is that tight, maybe they're not going to be able to iterate on that too much.
Right. I was wondering, John, if you know if this is a population, CDK4/6 relapse population, is the one that Pfizer is targeting, or they're just doing this as a stepstone to get into frontline patients?
I think it's certainly the place to start. The patient populations in frontline are going to be slightly different, and I do know that there are some interesting combo strategies that other players are looking at for these more selective CDKs. So I wouldn't put it past Pfizer to come out with strategies in the front line that are distinct from the Ibrance strategies there. That said, I would view this as a real population, certainly, right? I don't think they're going to ignore later populations, especially post-Ibrance setting, given how many patients there are there. It's worth saying they also reported some CDK2 data, right? This is not the only selective CDK inhibitor that Pfizer has in development. And the CDK2, which has its own interesting combo strategies that you could do with it, also has 20% plus ORR, so it's not dead in the setting either. So they do have two good options here that they could pursue in sequence or in different combinations.
Right. I think this also reminds me of Pfizer's broader kind of oncology strategy here, because I've covered these medical meetings for a few years, and Pfizer is obviously missing from some of the at least impactful data readouts. So where do you see, apart from the ongoing Seagen buyout, obviously, where do you see Pfizer is this CDK space something that Pfizer could continue to do well in, given its legacy here, or is there something somewhere else?
Well, I mean, obviously, Ibrance has been a huge product for them, and they've really done a ton of development in this space. So I don't think that, Novartis' successes aside, I don't think Pfizer is going to lose their footing in CDK or cytostatics in general. They clearly have a strong pipeline there as well. Again, it'll really come down to where the tolerability is and what the combos look like as we start getting to bigger and more fulsome combinations in the first line. You mentioned the Seagen acquisition, which obviously has its own complications, but you're starting to see both Ibrance and Seagen going into first line, competitive data sets there. There's that problem for Seagen is another abstract, which I think we don't have on the list here, but the previously leaked abstract from the NCI study showing that IO outperforms Etcetras in first line Hodgkin. Right. So there's clearly some threats on multiple fronts to both Pfizer's established CDK franchises and to the established Seagen franchises. But you can't count them out. But they might be limited in market share as some of these other things come.
Right. Seagen and Pfizer. That's worth another entire webinar.
It's a whole other.
Right. Yeah. Okay. Let's
so before you guys move on, this part actually need make the call. Do you want to give it sounds like somewhere between positive neutral and neutral? John, what's your call?
I would give it a positive neutral. I think this was good data. It might not be sufficient on its own, but I think it's hard to read this abstract as well.
All right, that's all I need. Please continue.
Okay. The bispecific and the WEE inhibitor, I think, John, that's all you. Less
Sure.
familiar with those assets, except for Carvykti, of course. Go ahead.
We'll move pretty rapidly here. The bispecifics are very interesting here. We know that BCMA CAR-T outperforms BCMA bispecific pretty dramatically. The reason BCMA bispecific is interesting at all is because nobody can manufacture CAR-Ts, at least in sufficient quantity to dose the population. Legend, J&J, even Bristol say that manufacturing capacity is going to ramp dramatically over the coming years. So does that mean that BCMA bispecifics are going to have a lesser and lesser role to play in myeloma? I would have said that's fair to say. Not that the drugs aren't active, but other BCMA mechanisms are just so active that it sort of outshines them. GPRC5D is obviously another very interesting target, not approved yet, but a very interesting early target in myeloma, showing in CAR-T settings, showing almost equivalent efficacy to the BCMAs. So clearly also in that same highly active regime. It's a very reasonable question. If you could combine the bispecifics and get CAR-T competitive efficacy, and I think it clearly looks like they're getting there. They're 90% plus ORR, where they ought to be relative to CAR-T. Obviously, we don't know about durability, and that is an issue for the BCMA bispecific. But if you're getting to those highly active ORRs, I would be inclined to say there's definitely a role for you. But safety here, really, really an issue. Obviously, seeing DLTs at dose level 1, starting to see dose interruptions and reductions even at very low doses. And the CAR-Ts, for all historically, we've been very worried about their tox. Modern treatment of CRS and modern monitoring for ICANS is very effective, and the CAR-Ts don't seem to be overwhelmingly toxic. Certainly not the sort of aggressive CRS or epitheliotox that you're seeing in these combinations here. So I do think that the efficacy more or less hit the bar that they needed to hit, but I don't think safety is there. Infection risk, the CRS risk is just a little bit too high for this combination, at least as it stands. Not in this abstract, but it's also worth pointing out that J&J is running a collaboration with SpringWorks on their BCMA CD3 plus SpringWorks' small molecule, which also gets to 90+% ORR, and also has DLTs in dose level 1 and has some safety liabilities. So I guess the BCMAs are just not, the BCMA bispecifics are just not combinable, maybe, or you have to be very careful about patient selection, maybe. Anyway, it seemed to me that this is a tough one to bet on at this point.
Right, John, just on the safety side of CAR-T, just wanted to get your opinion on this emerging toxicity, neurotoxicity of Parkinson's disease. Do you think this is a big problem for the CAR-Ts right now? Because I've heard some mentioning from doctors.
Yeah, a big problem. What's the magnitude of the problem is the real thing. Is it an issue? Yeah, I think all of the CAR-Ts come with CNS liabilities. And clearly that makes patient selection very, well, not challenging, but you have to be careful, right? You can't be giving these products to patients that have existing or overlapping liability. That said, and so maybe there's a niche for a bispecific that has lower CNS tox issues. Not that I think that really is the case for BCMA plus GPRC5D. So maybe that's a niche for something else to fill, but I don't think it's going to blunt momentum for the BCMA CAR-Ts. Certainly, we still see more than adequate demand for the products. The Legend, I mean, the easy way to look at this is just to say, look at the market share between Legend and Bristol compared to the data, right? Legend data is so much better than Abecma data. And market share is still very split. Clearly, the limitation here is manufacturing, not the actual clinical profile of the drugs.
Yeah, makes sense. Now moving on to the WEE inhibitor, John.
Yeah, the WEE inhibitor. This is a target that seemed to be getting a lot of attention 2 years ago, year and a half ago, maybe, and then it And then it really dropped off the radar once early data looked a little disappointing, and I think you saw this from Novartis as well. So Zentalis with the subgroup showing meaningful ORR here is good. It's a refractory population. It's a biomarker-defined subgroup. So all of these are positive signs for the ability to find a niche. There are combos for WEE1 that seem reasonable. That seem like a reasonable hypotheses as well. That said, I would say that while the abstract on its face looks good, I'm not sure it was sufficient to move markets here. Really, obviously Zentalis reacted pretty well, but I see it giving back a lot of that. And certainly most of the other WEE1 competitors, other people in the space, are not seeing the sort of read across from this abstract that other TIGIT players were seeing from Morpheus Sliver, right? You saw Arcus, for instance, up 26% on this Roche abstract. You don't see anything like that in the WEE1 space where the other players beyond Zentalis are getting such a big boost. So
I know we're sort of close to time, but I want to get your opinion, John, on another ovarian cancer data set that we already see in the ImmunoGen, the EDC data. Do you think that kind of market movement is warranted just based on the data we've seen from
the ImmunoGen data set is great, and I think right now there's not really much of anything challenging. So I understand where the market movement came from there, and I don't think that you're not going to see head-to-head comps versus WEE1 in a biomarker selected population. Maybe there's a niche, but the ImmunoGen data also biomarker selected also seems very, very strong in its population.
But is there any additional data set that you would be looking for, like see in the ASCO, definitely in the ASCO presentation? I mean, we already have the headline data.
We already have the headline. I mean, look, the questions for any ADC program are always going to be the safety and what proportion of the population is going to tolerate treatment and what discontinuations look like. So none of that is going to certainly we're going to be paying attention to that. I don't think any of that is going to come as too much of a surprise, just based on the body language management. But I think we know the headline there, and I don't expect that you're going to see. Well, certainly the stock reaction is not going to be as dramatic on the final data as it was.
Got it. All right. Leon, I think we finished our part.
Yeah,
absolutely. Let's we'll pass it off to.
That's a fantastic discussion. I hope you agree. My grade on your pretty much summarize what you said. But thank you so much, John. You feel free to stay, but I understand you have a family duty, and also this is an institutional investor, kind of a cracking finally cramming season. I understand you probably want to catch some sleep because ASCO is coming. Going to be really very little time to sleep. If you need to go, please feel free. But anyway, in the meantime. Yeah, thank you so much, John. And Angus, I know you're going to stay with me. So we're going to move to the next session. In the very beginning, Lee was a little bit late, so I can introduce her before this session. So Lee's she's veteran of a drug developer, and she has been around for a long time, from like big pharma, and then go to biotech, starting as a first a few employee of Apellion until Apellion today become one of from running next generation immuno-oncology company with this very interesting technology that target glycan biology. And their scientific founder, Karen Bertozzi, as you know, from Stanford, just won Nobel Prize last year, exactly based on technology chemistry that she used to develop the current platform on Apellion. So that's about Dr. Li and Dr. Peng Li. So I'm very happy to have her here today to share the panel with me to discuss the second session, which is the rise of Asian biotech. I want to clarify that when we say Asian biotech, we're not just talking about a biotech with headquarters in Asia. We're talking about biotech really pioneered, or the key drivers are Asians, ethnic Asians, could be Asian Americans, Asian Chinese, Asian Singaporean, Asian Hong Kong. You get my point. With that being said, let's just dive in, I will start with the first one, the late breaking abstract 106, which is CARVYKTI. It's actually a quite interesting story. We are not supposed to know the data, but we knew it because, fortunately or unfortunately, our academic conference organizer, EHA, European Hematology Association, they were supposed to report this data, and they were supposed to put in embargo, but somehow they leaked it. And in April, so data came out, and that was really good. And as you can see here, this is definitely background CARVYKTI, as you know, so far as just John and Angus just talked about. It is a best-in-class BCMA targeted CAR T, and was originally developed actually in China made by Dr. Fan Xiaohu. I think he's in the audience as well. Thank you very much. Then this drug has been, kind of I would say change the paradigm of the multiple myeloma treatment in the past couple of years. We already saw the initially 100 ORR was presented at ASCO 2017. People had a little bit doubt, but turned out it was the case because the pivotal trial turned out to be 98% ORR. And now they moved to second line. This is second line. That's why CARVYKTI, CARTITUDE-4 is so important. So in this phase 3 second line multiple myeloma study, they show 74% reduction of disease progression. Just give a little bit context, as you know, Abicma from BMS also reported the result in similar, not identical setting, have 51% reduction. So this is clearly best in class so far in BCMA space. And some analysts and also investors, even some biopharma and biotech executives argue that because the data is so good, that make the other experimental drug in this space, especially those BCMA targeted therapy, really have a hard time to, I would say, meet the bar right here. Of course, there's a little bit caveat, as Angus and John already mentioned, that even the CARVYKTI turned out to be the best in class, but their sales not there. Their sales pretty much on par with Abicma, despite the inferior data from Abicma, just because the bottleneck manufacturing. However, based on my understanding, J&J and Legend has been working this really hard. And recently, as Angus reported himself, actually, J&J already took some manufacturing capability, I believe, from Novartis, correct me. So I guess the bottleneck gonna be still there, but gonna be a little bit less constrained than right now. So I guess that would be really important. So I guess the bottom lines for those experimental drugs should not bet on the J&J and Legend would always have the bottleneck manufacturing, especially the data is so good. So with that being said, that's what I can tell about this abstract. I know there's probably something, Angus, you want to add, please feel free.
Yeah, I think, Leon, what I would say is of all the positive things we've already discussed, I think there is a question of where the ceiling of CARVYKTI really is, or whether we have reached that point at this point, because we have seen that, I mean, in late line therapy, nearly 100% ORR, there's no way you could top that beyond that. So how does that going to translate into even earlier treatment settings? This is something that we'll be keeping a close eye on moving forward, how CARVYKTI performs as we go into earlier treatment lines.
Correct. Yeah, so I don't know after this data whether people adjust their peak sales. I remember at JP Morgan, J&J already said they expect the CARVYKTI peak sale at least $5 billion. I guess after this data set, they probably can go even higher.
Yeah, and Abicma is 1 billion. BMS estimate is 1 billion.
Yes, I know. I mean, that's kind of interesting, right? People already know based on data, the peak sale going to be 5 times difference. But if you look at the current sale, they are kind of comparable. I just really tell you the CAR T is a totally different beast compared to traditional biologics, a small molecule. You really have to plan out your CMC and your CDMO carefully. Yeah. It's not like I would say it's not easy for small biotech to play. I remember we did an analysis with the closed door discussion. We asked those insiders what do they think the most I would say commercially attractive cell therapy company, and top 3 actually all big pharma / biotech combination. Not even one biotech made the cut. I mean, because if you really have worked on this before, you will realize the capex is just so high. You have to invest so much money. A couple billion to get started, right? It's way beyond just like a clinical trial and to get an FDA approval. So that's what I can say. Anything else you want to add before I move on? I guess this is a clear positive.
Obviously clear positive.
All right, so I'm gonna move to the next one. This is actually another Asian biotech. Again, this is a Gracell, listed in the NASDAQ, but they have office both in China and the United States. I believe they already started some clinical trial in the United States. But this trial is actually, this data come from their China clinical trial. I just chatted with their executive offline before I came to this webinar, but everything is publicly available. Nothing internal, like inside information. So don't expect anything special from me. I just summarize, the some information probably we don't say that from the abstract. So obviously, people may ask, look at this. Okay, yeah, like Angus just said, his voice is still there. Cavacti achieved 100% ORR. And here you only have a 93%. You would argue this pretty much on par with the Bluebird data, BMS data, which is a BICMA, which actually was 24% when they presented data in 2017 ASCO, where Angus and I actually happened to be in the audience at the same time. We just didn't know each other. So this looks like that. You may argue, so what's special about this? How could this be kind of differentiated from Cavacti? So based on my conversation with the management, they point out a few things. First of all, they believe the patient actually in this trial a little bit sicker compared to in the initial trial from J&J and Legend, which is, I believe, their pivotal trial is a CARTITUDE one. That's number one. Number two, they also want to emphasize that they are manufacturing actually based on this fast T CAR-T technology. As you know, that's a big news from ASH last year. This whole idea is like they can manufacture the cells much faster, and they're supposed to have a higher percentage of T stem cell, what's the name? Stem cell-like memory T cell phenotype. The portion is going to be higher. And based on the current understanding in academia research, people believe that if you have a higher percentage of TSCM, you would have I would say more effective T cell in terms of killing, but also have a lower toxicity, which is sort of reflected in their initial toxicity data. As you can see here, their CRS is pretty low. Actually, they did not report any neuron tox. So ICONS is the acronym for neuron tox. So that's kind of interesting. But I guess we just have to keep our eye open, look at more details. I mean, again, there's always a caveat when you do cross trial comparison, because the patient population unfortunately will never be identical. But I think, in my opinion, this is kind of I'm cautiously optimistic. I will get a positive neutral. That's my opinion. So anything else, Angus or Pengli, you want to add before we move on to the next one?
Nothing from me.
All right, so I'm gonna hand over to Dr. Peng. I know you're gonna talk about the following 3 bullet points. Please take it from me.
Okay, thanks, Liang. From Sichuan Baili Pharmaceutical, there were 2 abstracts, both related to the targets, EGFR and HER3. The first abstract, 3001, is about a first-in-class bispecific ADC. The design of the drug, it's 2 different targets, bispecific, with a topoisomerase 1 inhibitor, and through a cleavable linker. And this study was a phase 1 study to demonstrate the safety and also showing the efficacy signal. And they demonstrated it's very well tolerated. MTD was determined to be 3 mg per kg for dosing day 1, day 8, and every 3 weeks. And if you're dosing every 3 weeks, just day 1, it can be 6 mg per kg. One thing worth to mention, there's no interstitial lung disease from the trial of over 100 patients. Lastly, the trial demonstrated very encouraging efficacy data, particularly in the heavily pretreated metastatically advanced non-small cell lung cancer patients with the EGFR mutation. They showed a very encouraging of ORRs of 61.8%. So overall, I think it's a very encouraging trial with a first-in-class drug. Additionally, they also reported another abstract, which is antibody targeting EGFR and HER3 without the ADC conjugate. And in this trial, they compared the monotherapy versus combination with paclitaxel or doxil. And very well tolerated, slightly better safety margin than the ADC. As we all know, naked antibody typically have better safety than ADC for the same targets. And here they compared the dose of 12 mg per kg for the combo versus the mono of 16 mg per kg. And dosing was every week. And very encouragingly, the combo showed better ORRs than the mono. So I think it's quite encouraging, and with the potential to be a first-in-class drugs.
Great. So Dr. Peng, I actually have a question for you in terms of this EGFR HER2 bispecific ADC. Just probably one tidy correction. They actually got from 2 different companies. I know they're confusing. So one's Sichuan Baili, another, I believe, let me check the name, it's a VP. It's definitely from different companies. That happened, that's actually another interesting stuff. There's a lot of converging innovation, especially among the China ADC companies. I guess you and I both agree that there's a lot of innovation in ADC space among the Asian biotech companies, especially those in China. So this is a very good example. I think I have not seen other bispecific ADC yet against this target. As we know, there's EGFR and c-MET bispecific, but they are just like Valina bispecific, right? There's no payload attached to it. Actually, one thing I wanted to get your opinion, because some people argue those EGFR HER2 bispecific, this naked antibody by itself may already have some efficacy. So how much could we really assign this pretty impressive OR to the monoclonal antibody itself based on ADCC or ADPC effects, rather than because the target delivery of a payload? What's your opinion? What's your view on that?
Yeah, that's a great question. And when I looked at the abstract, I was also curious, the 2 abstracts, whether they're from the same company or different company, and how to weigh or how to compare the same targets, naked antibody versus ADC. It's interesting, both studies were sponsored by the pharmaceutical company, but it's possible they were developed by different companies. So I think to compare the efficacy, by comparing those 2 abstracts, it's hard to conclude because both trials are in different indications, different tumor types. But in either indication, definitely the efficacy signal, I think it's very encouraging for both molecules. In terms of safety profile, definitely the ADC modality, the molecule has a narrow therapeutic index compared to the naked antibody. I think if the company has if a company has a similar molecules, probably it's worth to do a small trial just to compare the therapeutic index and to before doing a larger trials. But overall, I think it's very hard to prioritize one versus the other just based on limited information.
Yeah, I guess that's always a challenge, right? This kind of reminded me about the HER2 ADC or HER2 monoclonal antibody space. As you can see, the Herceptin or trastuzumab as a backbone, you have several drugs approved based on it, right? You have a Herceptin, which is like a naked monoclonal antibody been around for almost 20 years. And you have the first generation of ADC, the TDM1. Now you have the best-in-class in HER2, and I have a bunch of others claim to kind of beat in HER2 in some way. And they're all based on the same monoclonal antibody, more or less the same. And but exactly like you said, when you make the drug more potent, their therapeutic window unfortunately also becomes smaller. Once that happens, actually, they limit their application to the low risk patient. I guess right now, even for HER2 move to front line, it's not that straightforward, right? I mean, we just talked about ILD, because the current standard of care for frontline HER2 positive breast cancer is I would say, studied and understood. And just you bring in HER2, despite the really tremendously improved efficacy, the tox profile just not as good as the current standard of care. I think that's something probably we should think about it. Probably there's some merit, like you just Dr. Peng pointed out. There's two kind of a similar assay, but with a slightly different design. They provide different applications. So that's my plugin. So, but I want to ask you whether you should assign that as a positive, neutral, very positive, or something else. You think that the green color is justified, or you think you want to tone down a little bit? Up to you.
Yeah, I would suggest positive, neutral. Okay,
all right. Please continue.
Okay, so the abstract 3023 is a HER2 ADC. And this is developed by, Duality. Actually, their chiefs are in the audience. Okay. Okay, that gives me a lot of pressure then. Don't worry about it. They are all good friends. So I would just go over the background of the molecule. So this is a humanized anti-HER2 IgG1 monoclonal antibody linked to also a DNA topo isomerase 1 inhibitor. But looks like this is a proprietary topo isomerase 1 inhibitor from Duality. And the drug to antibody ratio is also similar to in HER2, around 8. And this is a phase 1 trial, phase 1/2A trial to demonstrate safety and also efficacy signals. The dosing regimen is similar to in HER2 as well, every 3 weeks. And this trial did a dose escalation of 6 levels from 2.2 all the way to 10 mg/kg. And it's very well tolerated. And the safety profiles, to my collection, is very similar to in HER2. Very well tolerated. And the half-life of the drug is around 6 to 7 days for the dose around 6 to 8 mg/kg, which I think is also similar to in HER2. And the drug demonstrated a very encouraging, compelling efficacy. About 44% of partial tumor response. And also among all the patients, the disease control rate was around 88.5%. So overall, this drug is very well tolerated and with very encouraging anti-tumor data.
Yeah, so very nice summary. I just want to add a little bit color. So in case you people didn't realize which drug this is, actually, if I mentioned the 170 million upfront cash between Duality and the BioNTech, probably rings some bell. Yes, that's the drug. So if you recall early this year, there was one of the biggest transaction, our license deal between the China biotech and overseas company. That's this one. So they got that 170 million US dollar upfront, and the total is, I believe, is around 2 billion total value. And at that time, actually, people were surprised. How come Duality? We never heard about this company, still private. How did they make this happen? So actually, like I said, I know their CEO, SVP of BD, as well as their CSO fairly well. I get a little bit information from them, which is by the was public. By the time they cut the deal with the BioNTech, they already dosed around 40 patients in the United States. So that's very important. I want to point it out. That's why, in this abstract, we mentioned highly, this is a global multi-center first-in-human trial. It's not a China-only trial. So the data actually a lot of them come from the United States, number one. Number two, a lot of patients heavily treated. As you can see here, the median line is 7 prior line. And actually 1/3 of patients were treated by HER2 ADC already. So that's very important. So this is why people say, wow, this is a 44% OR. It's not as good as in HER2. No. You have to look at the nuances. The patient characteristic quite different from in HER2, which is, I believe it's Destiny Breast 01. That's a pivotal trial for HER2's approval in 3 line, per line and above. So this is quite different. Another thing I think I want to point it out is that they dose up to 10 milligram per kg. That's quite impressive. Because in HER2's dose actually is either 5.6 milligram per kg or 6.4 milligram per kg. Actually, in breast cancer, I believe they are 5.6 milligram per kg. So as you can see here, it's a lot higher. I believe when I talked with them, one reason BioNTech took this one because they thought this could apply to patients who progressed in HER2. So if that's the case, if the data really hold true, I think it would be very, very interesting. But again, now we know why BioNTech paid $170 million US for this asset along with another one. So that's my 2 cents with this one. So I will let Dr. Peng decide whether they are positive or positive neutral. It's up to you.
Yeah, definitely positive.
Okay, thank you. Do you want to continue?
Sure. Yeah, so the abstract 1155.9 is about novel drug against CSF or colony stimulating factor 1 CSF1R receptor, which is an antagonist with very good selectivity with minimal inhibition of c-KIT and the PDGFR. This drug has been granted a breakthrough therapy designation for tenosynovial giant cell tumor from FDA in January 2023. The drug was developed by ASCO Therapeutics. And this trial is a phase 1B update, particularly focused on the efficacy. It demonstrates very, very good signal. The ORR was around 75% for including 2 CRs in the high dose of 50 mg daily. And also 40% of ORRs at the lower dose, 25 mg. And so overall, I think it's very encouraging. I mean, F1R, it's a challenging target with good anti-tumor activity observed in this rare tumor, like joint tumor type, but had a lot of failures in other clinical trials. I think seeing this drug with such a impressive data also in the joint tumor, it's very encouraging. From my perspective, if after continuous clinical development, if this drug can apply to other tumor types, that would be very encouraging.
Yeah, so I want to share probably a little bit additional color about this one. I think for me, the biggest point for this one is, their OR really a lot better than the current standard of care, which is Daiichi paxitinib. That was approved actually during my time as a post on the Memorial Sloan Kettering. The pivotal trial was done over there, so I'm rather familiar with that one, fortunately. So that's one. I guess, the data numerically look right now, again, the caveat from cross-trial comparison definitely look better than the other one. But, again, this is, I believe, is a China-only trial. The patient population probably a little bit different from the other pivotal trial, which obviously is a multi-center global trial. As we discussed, unfortunately, the phase 2 data are always the best data. And once you expand to the pivotal or phase 3, the good data always, good efficacy will drop a little bit. But I'm cautiously optimistic based on this. And also offline conversation with the management, they confirm that actually their toxicity profiles are very good, which is important, as you probably know. If you don't know, the tenosynovial giant cell tumor (TGCT) is a kind of not super devastated cancer. They don't kill patient very fast. So the good tox profile is very important for the patient to use it for a long time. That's actually something I would be very curious to say. Obviously, just abstract. I think Dr. Peng and I are gonna be really pay attention to their nuances, because that's really make a huge difference. So for me, I probably would give a positive neutral. With the, that's just me. I don't know whether Dr. Peng you agree or not.
Yeah, I would give positive.
Okay, I'll go for you because you are the lead. All right, so I will cover last one real because I know we're running a little bit behind time. This is real quick, actually. I actually evaluated this asset before, so I know this a little bit more than regular people, I guess. So this is a zorefertinib originally developed by AstraZeneca, and Alpha Biopharma bought their China rights and then they developed them. So simply speaking, this drug is a kind of I would say brain penetrating version of gefitinib. So you more or less put into the first generation of EGFR inhibitor, but they have a really impressive brain penetrating capability. That's why they have this data, as you can see here. However, I want to bring your attention right here is the right now standard of care changed. Standard of care is no more gefitinib, at least outside of China. It's not gefitinib or inotinib anymore. It's osimertinib. Osimertinib by design also has a decent brain penetrating capability. So what I really want to say is yes, the data looks very good. Compared to the old standard of care, but how does it compare to the current standard of care? Actually, I don't know. Again, this is impossible to do cross-trial comparison. As you know, the first generation of an EGFR inhibitor, they did not address all the different mutations of EGFR, while the newer generation like osimertinib or furmonertinib and alamertinib, they all have some improvement over the first generation. So for me, I would give it probably something like a positive and neutral. By the way, the PI believe the study PI is Yilong Wu, one of the most famous lung cancer doctors. So I guess if a Dr. Wu really, kind of advocate this to try, probably there's something else. So I will put those together, I would give like a positive neutral. That's what I would assign to this abstract. Despite, this looks like an impressive headline, but I just want to put a little bit caveat, the background of this one. With that being said, I know we already run out of time with this session. Thank you very much, Dr. Pong. I know you're gonna stick around, so let's just hand the baton to Dr. Park. Could you please take over, and Angelis and I gonna try to play the second fiddle with you.
Oh yes, yes, great talk. Really enjoyed listening to you guys, and I was going to just highlight some of the practice-changing trials that kind of just highlight over and see what the data is going to look like. So the first study is the Indigo study. This is a very interesting study because it takes into account two things: one, the IDH inhibitor, and two, this is a very unmet need patient population. So Indigo study is a global randomized double-blinded phase 3 study of vorasidenib versus placebo in patients with residual or recurrent grade 2 glioma with an IDH1 or IDH2 mutation status. And so just to kind of give everybody a quick background, IDH1, IDH2 inhibition is something you may have heard in a cancer called acute myeloid leukemia and AML. And so here in America, we have FDA approval for this medication for patients with IDH1 or IDH2 mutation. Well, it turns out that for patients with glioma, 60 to 80% of these patients have an IDH1, IDH2 mutation. And so in this study, it's a phase 3 study. Patients are randomized, with WHO criteria, with mutation status. It's actually patients that received one or more surgery for glioma within 5 years and had confirmed enhancing lesion on MRI. And so if patients had that and proceed with treatment with chemotherapy, these patients tend to do well, but if they recur, they tend to be very chemo-resistant and radio-resistant. So that's where this unmet need is. So in this study that's going to be released at the plenary session, this is going to be very interesting in terms of targetable medications for brain cancers with IDH1, IDH2 mutation. And this is about 5 to 10% of all brain cancers. So Leon, back to you. Any questions?
No, you covered really well. So you want to give the super positive? That's what you are saying?
Yeah, super positive. I think this is something when patients recur, it's an unmet need. So I just have to look at the DFS and so, but it looks really good.
I have one question,
Okay.
Dr. Park. Just how prevalent is IDH screening in brain cancer patients these days?
Yeah, so we do it at all of our cancers here at Norton Cancer Institute, but it's standard of care. The big issue is the grade 4s, the glioblastoma multiforme. The IDH1, IDH2 mutation is very low. It's about the 5 to 10%. But in these low-grade gliomas, it's about 60, 80%. So everybody should be tested.
Oh, I see. Got it. Just some background for this study. Actually, this is a drug by Servier. It got the drug from acquiring Agios oncology portfolio a few years ago, and it also includes Tibsovo, ivosidenib, which is an IDH1 inhibitor as well. So just some additional background on this study. Dr. Park, to you next, the second study.
Yes, thank you so much. So the second study is called a prospect study. And a prospect study is a randomized phase 3 trial of neoadjuvant chemoradiation versus neoadjuvant FOLFOX chemotherapy with selective use of chemoradiation, followed by total mesorectal excision for treatment of locally advanced rectal cancer. Now, this is a very interesting study because treatments for stage 2, stage 3 rectal cancer has changed in the last 4 years. This field is evolving fast. About 4 years ago, the standard of care treatment was chemoradiation, then you do surgery, and then you do chemotherapy. But however, the toxicity of the radiation was very tough on our patients. And also the surgeons also mentioned having radiation before surgery is very hard for the resection. And not many people would like to have chemotherapy after having surgery. And so one of the developments a couple of years ago at ASCO was for patients with this, they started talking about let's see if we can do all the treatments before the surgery, which makes it easier for our patients. And so the study that became the new standard of care was called the phase 3 Protege study. So in the phase 3 Protege study, they had all the treatments at the beginning. And so it's called a TNT, total neoadjuvant treatment. And so in this study, the patients received neoadjuvant chemotherapy, followed by chemoradiotherapy, and then did surgery. This became the new standard of care with much longer disease-free survival. 3-year disease survival was 76% versus 69%. And so that became the new standard of care. And so building upon that, that's where this study comes into play at this year's ASCO. And that is, can we just get rid of radiation completely? And so in this study, what they did is the neoadjuvant FOLFOX chemotherapy with the selective use of chemoradiation is the business end of that study. And so what they did in this study is they did the FOLFOX chemotherapy, and then they evaluated the patient. And if the patient had a greater than 20% reduction, they would not receive radiation at all. And so that's something that's very exciting for our patients. Now, if they did have, less than 20%, they did get the radiation. So this is very interesting because we might not need radiation therapy for our patients with stage 2, stage 3 rectal cancer. Back to you, Leon.
Yeah, thank you very much. So I guess that's another super positive. That's another like practically changing one. Okay, please continue. I'm learning a lot.
Okay, so the next study is we got the LAB3, which is probably the most eagerly anticipated plenary session. There was a lot of debates on this. And so just to kind of give everybody a quick background, osimertinib, based upon the FLORA study, became the standard of care for stage 4 EGFR mutant lung adenocarcinomas. Because of the great response rate, it became earlier and earlier lines of treatment. And one of the earlier lines of treatment is adjuvant study. And so in this study, in the ADORA study, not the FLORA was stage 4, but ADORA study, they finally discussed the overall survival analysis of adjuvant osimertinib in patients with resected EGFR mutated stage 1B, which is a very small patient population. By definition, this means it's greater than 1 centimeter, but less than 2 centimeter. So that's a very small patient population, up to 3A. And so this is a very wide range group. And so in this study, what they found, what they're going to find is that EGFR targeted therapy is going to show some overall survival. Now the question is how much, okay? And so to give everybody a quick background, in December 2020, the FDA approved osimertinib for use as adjuvant treatment based upon the DFS, and the overall survival is going to be finally released in this study. And so it's going to really boil down to, will 3 years of osimertinib, would that increase the financial toxicity? How big is this delta? What is going to be the p-value? What's going to be the hazard ratio? It's going to be definitely be a positive study, but the question is, how big is that delta?
Yeah, Dr. Park, just one question from me. If we look back at the original ADAURA PFS data, DFS data, the risk reduction was very impressive, 83% reduction. Just heading into this trial, what was your expectation, kind of the range of OS benefit that you would expect to see, and what would you think would be really practice-changing OS benefit?
Yeah, so in the overall median follow-up, it's going to be what, 44.2 months versus what, 19.6 months with the DFS at 65.8 versus 21.9. So that's a 43.9 month disease-free survival. So at least what, maybe 18 to 20 months. We'll have to see what it looks like.
Right.
But the key here is the toxicities of chemotherapy. I think that's the other thing we have to look at. From a doctor's perspective, it's all about the toxicities. This is well tolerated. Chemotherapy can be very tough. Some of these patients end up with permanent neurotoxicity with peripheral neuropathy that never recover from. So that's something else it's in the osimertinib's arm. Any other questions, Leon?
No, not at all. So, well, since like, there's going to be another super positive, in your opinion,
Yeah,
I'm going to just listen to you.
But I will tell you, this opens up Pandora's box. And so if you look at the NCCN guidelines for stage 4 lung cancers, I mean, it's all across the board. Like, so right now, if you have a stage 4 lung adenocarcinoma, we look for EGFR for osimertinib, KRAS G12C for second-line KRAS G12C medication, ALK rearrangement, ROS1, BRAF, NTRK, MET, RET, HER2, PDL. And so all of these things are going to go earlier and early lines of treatment. Now, the question is, are they all going to make it to the finish line? I question KRAS G12C in an adjuvant setting, but stuff like BRAF, stuff like HER2, I can see, especially with the TDXD data, I can see that slowly moving into the adjuvant space. Any other questions?
No, I mean, that's already gonna make my head spin. We're gonna make your job even more difficult. We're gonna keep pumping more, video drug. We're gonna dice the patient population even in smaller fragment. That's how we increase our chance to get approved. But I know actually sometimes you are practicing physicians, right? I guess you probably sometimes I know some of the lead, I would say, lay back oncologist told me, why you guys make our life so difficult right now, especially lung cancer, right? We don't, there's overlapping choices. NCCN, even like their, let's say, category 1 recommendation have a 2 or 3. So which one should I choose? I guess, that's really become art, right? Obviously your job security is very good. I don't want to take your job. That's too much thinking for me. But I guess that's anyway. That's just my my kind of admiration to what you do. With that being said, I'm gonna move on to the next one. This is something I will try to take a swing, but I'm pretty sure I'm gonna swing in the wrong way. Please correct me, Dr. Park, because I'm not a medical doctor, right? I'm a mouse doctor. So this one, the brentuximab vedotin, actually the treatment name probably is more well known. It's called Adcentris, I believe. That's a CD30 ADC from Seagen. That's actually Seagen's before passive came along. That I believe that's their best selling drug for a long time, and that's been there for a long time. And I guess the idea right here is really kind of we know ADC works, but ADC also have a pretty small therapeutic window, and also the toxicity, especially this is actually 6th generation of ADC. That means that their linkers not very stable, so their toxic profile is pretty bad. If you can replace it with a nivo, which is not, I would say, not a safer drug in comparison. I don't know the data yet. I guess the LBA have not released any data yet, but I guess the become LBA, late breaking abstract must have a really stellar data. So I'm just guessing this kind of a trial would really change the practice if it really work. I guess the efficacy probably going to be better. Otherwise, it would not be selected. And also, I assume based on my understanding of Nivo's clinical profile versus another 2nd generation ADC could be better. If that's the case, if you look at the combination, one is NAVD, another is BVMVD. I guess my guess is NAVD can have a lot better tox profile. If that's the case, understandable. The patient and also physician would prefer the newer version, which is NAVD. So that's my guess. But I know actually nivolumab already approved in different settings for classic Hodgkin's lymphoma. I guess this is just a great expansion of nivolumab into as one of I would say cornerstone for classic Hodgkin's lymphoma. That's my best try. So Dr. Park, please correct me if I make any sense. If not, please.
I think you did a great job, and I love how you explained that. One of the things that I think about as a cancer doctor is, like you mentioned, the toxicity. 90% of these patients get cured. Okay, we used to cure them with a treatment called BCOP. BCOP is all of the strongest chemotherapy combinations you can think of, and the patients just get really sick. And that's how they did in the 80s and 90s. And then the next standard of care became ABVD, right? ABVD, but the B, you still had that bleomycin, end up with the pulmonary toxicity, interstitial lung disease. And so then we had the echelon study with the stage, with the phase 3 study that showed improvement in overall survival, but the brentuximab vedotin also has some patients that you have to keep an eye on, and are the patients with diabetes with peripheral neuropathy, because the peripheral neuropathy for brentuximab was actually stronger than the ABVD arm. And so ABVD, AAVD is considered the standard of care. And so now we have a new challenger, and this is checkpoint inhibitor. This is rose all the way up the ranks, third line, post-transplant, second line. There's actually studies with Nivo and brentuximab vedotin as a combination as chemo-free, and that's actually showing good efficacy in the second line space. And so now it's going to be at the top line, mono a mono, nivolumab with AVD versus brentuximab AVD. What I'm going to be looking for is biomarkers. Are they going to help us separate the two, not just the overall response rate? And secondly is the peripheral neuropathy of brentuximab. I agree with you. I haven't looked at the data, but it must be positive to get to this line. If it doesn't beat on all comers, I suspect there's going to be some patients with biomarkers that it would show improvement. So very exciting for our patient population.
Okay, so sounds like another green. All right, I'll do the same thing. Try to give my non-medical doctors a try with the last one. Actually, last one is really easy. FDA gave approval of this combination in this April based on this data. So I guess it's good. FDA already prove like a second line. Actually, this is Yescarta in second line, which is a relapse or refractory large B-cell lymphoma, big population. I think Gilead must be super happy. I just read their, while we're talking, I read their press release. So you can tell they're very happy. Actually, I know the PI, Frederick Locke. He was trained at Sloan Kettering, and right now he's a big name at Morphe. And he's a study PI. I had a fortune to talk to him. Very, very nice man and very knowledgeable. Absolutely, I would say, big shot in the CAR T space. He has led a lot of Yescarta trials and Morphe. And Morphe is a great place. So let's go back to actually what like Dr. Park and I discussed. Right now, actually, you really want to get a good trial done. You probably don't want to go to the the top 10 medical center. They are so competitive over there in terms of a really cool patient, like a Sarah Callan or like a Morphe. That's where you want to go. And I'm very happy. This one, Zuma 7, was led by kind of a, I would say, established PI from Morphe instead of from Sloan Kettering. So that's what I can say. The data is definitely good. There's no doubt. They already get approval. This FDA approved good data. So we are just need to learn what's the detail of it, why. All the analysts upgrade the sales forecast of Yescarta so much. Hand back to you, Dr. Park.
Yeah, so diffuse large B-cell lymphoma, 50% of these patients get cured. The standard care treatment is R-CHOP. Question, with the data that came out in ASH, we can use the ADC instead of R-CHOP, and I think that data is very strong. But then the question is, if the patients relapse in less than 12 months, that means those patients are in trouble. And so one of the first studies that they did was called the Zuma-1 trial, which involved patients with refractory large B-cell lymphoma that are in that trouble spot where they relapse in less than a year. Surprisingly, 83% of the patients had a response and 58% had a complete response, which is a very shocker in terms of the malignant heme community. With the median overall survival of 25.8 versus an overall survival of 43% at 5 years. So very strong data. Then the question is, what about the Zuma-7, the phase 3 study? And so that's where this study comes into play. Second line therapy, post first line chemotherapy for the high risk patients that progress after 12 months or before 12 months. And so this is going to be very interesting data. We're going to have to see what the overall survival here is. The standard of care treatment is the chemotherapy with autologous stem cell transplant. So this is a very good standard of treatment. So the only thing that this is going to do is highlight the lack of ability to get this CAR T-cell treatment, because right now with the industry slowdown, it's hard to get access to these medications. So I think that's what this is going to highlight. I'm very excited about this. We have to look at the data here on this one. Back to you, Leon.
Yeah, thank you very much. I think that's pretty much all we need to say today. And in the meantime, I want to make a correction. So what we said about the EGFR HER2, I'm correcting right now, actually it was a mistake. The assay from Sichuan Baili actually was a EGFR HER3. Thank you, the audience who corrected me. And my apologize. So that's actually means that these two assays actually they are different. They are actually have a different target. One is EGFR HER2, another is EGFR HER3. So I'm correcting here. You know, always nice if we made a mistake, just say it. Actually, we have a little bit time. Probably we're gonna take like two questions from the audience. If anyone want to have any question, please raise your hand. We can let you speak, or you can type in the Q&A sessions. I know we run out of time, but I guess we still want to give our audience to ask at least two burning questions. Please.
Hey, Leon, while we wait for the questions, I want to just like a quick overview of some of the market moving data that I see in my clinical practice. You know, I know with the Natalie study that you guys mentioned, I'm very excited about that. Abemaciclib, very strong data for you know, for adjuvant, but that diarrhea is very tough in clinical practice. Ribociclib, you have to watch the EKG. So it's going to be very interesting from a physician's perspective. I think most physicians prefer ribociclib because he's got the OS data over palbociclib in an adjuvant set in metastatic setting. And then the other thing that you mentioned is the ADC with these TDXD versus the TROP2. TROP2, you don't have that interstitial lung disease. And just to kind of remind everybody, when we do clinical studies, we look at grade 1, 2, 3, and 4. For interstitial lung disease, grade 1, for TDXD, patient have no symptoms. They have no shortness of breath. They have no coughing. The only way you're going to pick up grade 1, you know, interstitial lung disease is with a CT scan. That's what makes the TROP2 so fascinating. Back to you.
Yeah, thank you very much. So actually, I haven't seen any questions from the audience. If there's no more question, we're going to conclude today's webinar. Thank you, everyone. I know John already left. Thank you, Angus. Thank you, Dr. Peng. Thank you, Dr. Park. And also all the support from BioSpark, SAPA-GP, and all our median you know, collaborators from China. Thank you very much. This is a real team effort. A lot of people involved in our WeChat group have like over 20 people. So this is not something you think we just put it off on the ease. This is a lot of effort. So I would like to end with the, you know, the two quotes from our you know, our organization. First one is you know, the teamwork makes dream works, right? You can hashtag that one. That's number one. Number two, that's the motto of our In Science We Trust community, which I encourage everyone to follow us. Our motto is actually to travel far, travel together. That's pretty much, you know, well, you know, this probably is another about the Asian philosophy, right? We are probably, you know, we believe in individualism, but we also believe in collectivism, right? So we collectively together, we can advance ourselves. So finally, a little bit shameless plug-in. Since, you know, BioSpark and In Science We Trust community are the two lead organizer of this event. If you like what we do today, if you want to learn more about us, please follow us on LinkedIn, which is In Science We Trust community, or you follow up, follow with us, you know, BioSpark to, you know, by contact our BioSpark administrators or go to our website. I'll leave here for 30 seconds for the audience to scan the barcode to get in touch with us. You know, in the meantime, I wish everyone have a wonderful night, and I wish the audience from Asia have a wonderful day. And I guess many of the audience will go to ASCO. I'm looking forward to meeting with you and discussing with you. And we will also say whether we stand corrected from tonight's discussion. Thank you very much. You enjoy your day. Thank you.